Okay, everyone. I'm sorry I couldn't be with you today, but here we go. So if you look, this is our notes from how do non-cancer cells become cancer cells. And so we had already known that genes mutate, abnormal cells multiply, and we played a game with the healthy cell cycle, and we skipped some of these notes. So I wanted to go back and help you out with these. So a couple of things to notice. There was a typo in this section. You'll notice I added the letter C because that was not represented in our game, and we need that. We already know this one. So I wanted to make sure that was included, and I'm going to use just this little check mark. So I went into the stickers portion, and then you can kind of look around, and I found this back to school one, and I found this little that little check mark over there. So I'll be using those as well. Okay, so hopefully pause your video, get yourself set, and then we'll get rolling. So here we go. When existing cells die or damaged, other cells are triggered to grow larger. This part of our game was that G1 phase. So I'm going to label that one G1. And then it's kind of easy because you can use the game board. So we know that S came next, which stands for synthesis of DNA. And what we also saw was in between our G1 and our S phases was a checkpoint, right? We had to double check our cell to see if everything was ready. So right here, this represents a checkpoint. Where we had to double check to make sure we had enough resources gathered, that sort of thing. So the cell really does like take in nutrients and do all those things. After the S phase, notice how on our game board there was no checkpoint right here. There actually is really a checkpoint there. It's just not really well represented in the game. And that's another spot that P53 can act. Because it wasn't represented in our game, you don't have to, but if you wanted to, you could add another checkpoint actually right there. I think in future versions of the game, the instructors will modify that. After that was our G2 phase. So G2, the cell gets larger, it copies all the organelles, so it makes a double copy of everything. So first it focuses just on the DNA in here, and then it's focusing on all the other stuff that's inside the cell. So 6th grade, middle school, you learned about some of that stuff, but all the other bits and bobs it needs to be alive is grown there. And from our game, right at the end of G2, that's where we had another checkpoint. Oops, I guess I have to copy it first, plus. So we had another checkpoint right there at the end of the G2 phase, and that's really when we hit that M phase, and we had to roll things in a specific order because literally things happen in that specific order. So first off, the nucleus will dissolve, the chromosomes all line up, they get split down the center, and then they reach that last phase, and here's where that last one was left off, that cytokinesis phase. So at that cytokinesis phase, was the last checkpoint. And so that was our last checkpoint there, was right before that cytokinesis, so that we could double check to make sure that that cell was going to have what it needed. So at each of those points, the cell has a chance to say, oh, nope, not going to happen, and to go through apoptosis, my favorite word, apoptosis. So the G1 and G2 phases both have in common cell growth. That cell is really trying to grab all of the resources it needs. So it's cell growth and preparation for that DNA and for that duplicate copy to be made. The checkpoints really are to make sure the cell has duplicate chromosomes or DNA and all the resources it needs, right? So the S and the M phases of the cell cycle are critical to the two cells that result from the complete cell cycle because the S cycle makes sure that the DNA is duplicated and the M cycle makes sure that the cell itself is duplicated. What it means is you get two identical copies. Of that cell. So one of those copies would have been your button that you were using to move along. And the second one would have been if you got to the blue chip. So if you were able to make a blue chip in this round, and I just don't think we had enough time. If you would have had enough time, you could have gotten to those blue chips, right? Okay, so that's basically how the cell was supposed to work. So then we played again, right? And as I was playing, I got one red cancer cell, I got no blue healthy cells, and I had to start over 3 times. So we had to do a lot more of that this time, right? So there were lots of things that were different. You got extra rolls, right? There were times where you got to roll an extra turn. There were times that you got to jump ahead. There were times that you got to skip phases altogether. You didn't have to check. So you didn't have to use the checkpoints, right? You got to bypass those. What else did we see happen? We still had to, there were more start overs. I had to start over a lot more often. And I felt like it was much faster paced. Like it seemed to go a lot faster. The outcome of the game was we got more red chips, right? And we got more cells overall. Part of that was because you did have more time to play the game, but also part of it was because it was able to move faster and you were skipping forward. So P53 acted differently because it missed some errors. So that allowed the cell to skip some of those steps, right? It also had those cells didn't have all their resources, so it missed or bypassed resource needs. Like there were times I went all the way around and I maybe had one thing. I didn't have everything. So we heard Mr. Christian's talk in large group about how the cells are misshapen and they don't quite function quite correctly. And that's part of it. If it doesn't have all the resources it needs, then it can't just do the things. Okay. So how do non-cancer cells become cancer cells? Cells become cancer cells when the genes mutate and the cell cycle, it's supposed to say doesn't function normally. So the cell can skip checkpoints to continue the cell cycle. Even with errors. Okay. So when we have these P53 errors, that cell cycle kind of gets out of control. So I'm going to put that, actually, let's say cell cycle. Because it did feel a bit chaotic. Okay. What questions do we still need to answer to explain how non-cancer cells become cancer cells? Here's what I need you to do. I need you to go back to your original model and make edits. So what I'm going to have for you is you're going to go to Formative, and there's going to be a before question. So that screenshot you already have. So that's your cancer model screenshot, not the taco or your sandwich, okay? And then I want a now screenshot after you've made any edits, because now I want to see you add the pieces of the cell cycle and the checkpoints. If you want to include things like resources, you can. And one of the things I saw before was sometimes we just put random words on the page and we didn't show how things were related. So if you just have an arrow with some DNA pointing at something, that doesn't help us figure out how the cell cycle is related to that DNA, or how the checkpoints are related to that DNA. So somehow I want to see those edits made. So your new screenshot that you're going to add will be a now. So after your edits, so this one you already have. So that one's going to be the first one. And you're going to have a now update that I want to see as well. So the rest of this mod, now that you have these notes done, you are going to use the time to make your edits and then go upload that to Formative.